So first, what are protein post-translational modifications and what is the purpose of them? So we know that it came as a big surprise that our genome doesn't have as many genes as we thought. It actually has around 25,000 genes, and then we can increase this complexity of the genome through several different things. One is using alternative promoters, alternative splicing, and mRNA editing can then lead to many more transcripts. We can increase up to 4 times. However, the true and the huge increase comes with post-translational modification. You can take this combination of the proteins that are expressed by cells, and then by using different enzymes that modify them after the translation, you can actually increase the complexity that each cell, or the components of this thing that each cell can express. So there are many post-translational modification, and phosphorylation is that one we are going to be talking, and it can be on either tyrosine residue, as you can see in the A, or it can be on serine or threonine residues. But there are other post-translational modifications also important that we will not be covering, and I just want you to be aware of them. It's ubiquitination. We know that ubiquitination can target the substrate for degradation, but it can also signal different things, and it's usually at the lysine residues. And depending on how many ubiquitin residues, it will determine what the effect of ubiquitination is going to be. We have acetylation. Acetylation and methylation, both are most well known as the changes actually in the histones, because they can change how much your DNA is open to be regulated then for the gene expression. But the other proteins inside the cells, as it's shown here, actually can be acetylated and methylated as well. And there is also proline hydroxylation shown, that which regulates the activity of some transcription factor, which is shown in E here for HIF alpha, which regulates gene expressions for vascular cells, important for vascular cells. Now, the reason we are talking about is because the activation of protein kinase is really important, because it regulates many critical cellular processes. And again, here we have the top part that we have so far talked. We have GPCRs, or we have even the channels, and we have the production of second messengers such as calcium, CMP or IP3 and DAG, and also activation of small g proteins such as RAS and Rho. And then downstream from them, these, as we have talked, these either GPCRs or these secondary messengers can by themselves change the activity of different channels, but they also can downstream lead to change the activities of these kinases, which can feed back as well to them, so they can regulate now increase or decrease amount of these factors, but they will also be able to exert different effect on the cell well-being, or they're going to activate many different biochemical modification in the cells. They're also to change gene expression. They're going to also change proteins which are synthesized, and they will also be able to then modify the cytoskeleton components of the cells. And protein phosphorylation is mediated, so this is addition of the phosphate group and residues of certain amino acids inside of the proteins, and it's mediated by coordinated activities between protein kinases and phosphatases. So what protein kinases do, they take ATP and add that phosphate and make them phosphoprotein. And then what phosphatases do, they remove this phosphate as an inorganic phosphate and then leave not phosphorylated protein. So it's coordinated activities, depending how much effective protein kinase and protein phosphatase you have, will determine in the end how much of protein phosphorylation you are going to have in cells. Most commonly, we are going to be talking about the amino acids serine, threonine, tyrosine that are phosphorylated, and there are different kinases that phosphorylate serine, threonine, and the ones that phosphorylate tyrosine. What phosphorylation does, it can alter, so you're adding this negative charge on these amino acid residues, and this negative charge may alter conformation of this protein. And by altering the conformation, it can do several different things. It can change its function of that protein. It can either increase it or decrease the function. It can also change the interaction of this protein with other proteins. It can change its stability. It can increase or decrease its degradation. It can also open or close the ion channels. It can change conductance of ion channels. So by adding phosphorylation sites, you can alter the function of the target protein in many different ways. Apart from serine, threonine, and tyrosine, there are other amino acids that can be phosphorylated. Example is histidine, arginine, and lysine. However, the problem is that phosphorylation of these amino acids, for example, forms this both heat and acid label phosphoramidates bonds, and because they're so labeled, they're actually very quickly disintegrated and they're very hard to detect in conventional studies. So, which is represented here, it's really hard to see where histidine is being phosphorylated. Histidine is being phosphorylated in the protein because of its instability of this modification. However, we do know now there are certain the technical advances that have allowed us to actually fix those phosphohistidine, so to be able and develop the antibodies so you can detect them. And now there's increasing number of publications that are showing how phosphohistidine plays important role in cell signaling. For example, histidine and the beta gamma subunits can be phosphorylated and affect their activities. But again, they're much less known about it because only recently, as recently as 2 years ago, have the technical advances allowed us to be able to detect this more in our studies. So what we are now going to do, we are going to actually cover 3 major families of the kinases in a little bit more details. So you will know how they are regulated, and you will know approximately what are they regulated, what are their targets. So first we're going to talk about CMP dependent protein kinase, or pKa, otherwise known as pKa. So pKa is actually historically was the first protein kinase whose regulation has been found, and it has been found that it's regulated by CMP. So we know that adenylyl cyclases will catalyze CMP production after GPCRs that are linked to them. So we'll have increased amount of CMP. How does this lead to pKa activation? So pKa is tetradone. It's a tetramer. It has 2 regulatory and 2 catalytic subunits. And these 2 regulatory subunits are preventing catalytic subunits to be active. Actually, they're binding to their active sites, as you can see in the schematic here. So the red regulatory subunit has this little protrusion going into the blue catalytic subunit, and that protrusion or that imagination catalytic subunit is supposed to represent its active site. Now, when there's increase in CMP, you need 4 CMPs, 2 CMP molecules for each regulatory subunit. And when regulatory subunits bind the CMP, their affinity for catalytic subunit decreases, which is represented here as the change in the shape of the part that fits in a catalytic subunit. That allows catalytic subunits to dissociate from the regulatory subunit, and then they are active. So then they can add phosphosite at the serine or threonine that they are phosphorylated. So these are serine threonine kinase. So pKa serine threonine kinase. pKa is ubiquitously expressed, so it's all over the place, and it actually, I'm mentioning this many details about it because it will play a role in how it's regulated. So there are different genes for regulatory subunits, and there are different isoforms of it, and there are different genes for catalytic subunits. What's important to know that these regulatory subunits, not only they prevent the activity, but depending which regulatory subunit is expressed in that cell, will actually target pKa to different places. For example, regulatory subunit 2 is going to target pKa to the membrane. So if your cell is expressing regulatory subunit 2, you will have pKa targeted to the membrane. On the other hand, if you have regulatory subunit 1, they're mostly cytoplasmic, so you will not be targeting pKa to the membrane. Therefore, even though pKa may phosphorylate different targets, of which some are expressed on the membrane or close to the membrane, some in the cytosol, depending which regulatory subunits your cells are expressing, will determine which of these targets are going to actually, in reality, be phosphorylated by pKa. And also these subunits have different affinities for each other, which also will give a unique activation properties. For example, regulatory subunit beta, 1 beta binds CMP at a lower concentration than regulatory subunit 1 alpha. Therefore, if you have a regulatory subunit 1 beta, you are going to be activated faster than if you have regulatory subunit 1 alpha. So we have said that it's regulated by CMP and the calcium, because again, CMP is binding these inhibitory sites that prevent catalytic subunits to be active. Now, what is interesting about each of these kinases, or family of kinases we're going to be talking about, is that they're able to maintain their activation even when the signaling for them is lost, which allows them to be something called kinases. So they will need the initial signal to be activated, but then that signal can persist for much longer time. Their activity persists for much longer time than the signal that initiated their activation. And PK does this in three different ways. One is two different ways, sorry. It's that there is, with the persistent signaling, longer signaling it actually can lead to degradation of regulatory subunits through ubiquitin proteasome pathway, which will then, so if you don't have any regulatory subunits, it cannot inhibit catalytic subunits. So even when your CMP levels go down, there's not enough PK regulatory subunits to inhibit your catalytic subunits, so you will have maintained activation over longer times. Also, regulatory subunit can be phosphorylated actually by pKa catalytic subunit, and that phosphorylation is going to decrease the binding affinity of regulatory subunit to catalytic subunit. And finally, it can also lead to the changes in the expression levels of regulatory subunits. If you express less of regulatory subunits, you will have more of catalytic subunits, and therefore you will have enhanced or maintained activation of pKa. As I have mentioned earlier, that depending on which regulatory subunit you have will determine whether you're going to be targeted to the membrane or in the cytosol. This targeting or localization of pKa occurs through the group of proteins called A kinase anchoring proteins, or ACAPs. Now, ACAPs were originally isolated with pKa, and that's how they got the name. This family of proteins have three very important domains. One is pKa anchoring domain, which can be, so they have anchoring domains actually for many other enzymes and kinases and phosphatases. So they will have the anchoring domain for kinases and phosphatases. They have a localization signal that can target them to different places in membrane, in the cells, and you can see it here. They can be targeted to the membrane, but they can also be targeted to the nucleus. They can be targeted to the cytoskeleton, which is in here with microtubules. They can be targeted to the vesicles, and they can be also targeted to mitochondria. So there are different ACAPs that have target sequencing that take them to different spots. And then they also have these binding sites that will recruit protein kinases and phosphatases, for example, and they also have the ability to form these big complexes. So what ACAPs are really important is they form this network in which complex in which all other signaling molecules are going to be localized at the place where they should be. So for example, if you look at ACAP 79 or 150, it has domain that will allow it to interact with AMPA receptors, but it also binds PKC, P2B, which is phosphatase, called calcineurin as well, and PKA, and it allows all of them to be present close to the sites which they phosphorylate. So this allows this localization allows specific phosphorylation of the target protein that should be phosphorylated very quickly. It allows it to be very quick, and it also lowers the background, because you can imagine if you had pKa all over the cell, it will be able to phosphorylate different targets, therefore you have much higher background activity. What are the targets of pKa signaling? Here are some of them that are shown. One very important is, we will be spending much more time later talking, as change in gene expression through phosphorylation of CREB protein, which is short for CMP response binding element protein, which goes to the nucleus and can then lead to the activation of transcription. We have talked before that pKa regulates catecholamine synthesis. Remember when we talked the tyrosine hydroxylase, when it's phosphorylated by pKa, it can increase its activity. It also will change, it can phosphorylate AMPA receptors, and phosphorylation of AMPA receptors at serine 35 by pKa, and you have shown it here, localized right by the AMPA receptor with ACAP 79 we just talked. This phosphorylation actually sustains surface expression of AMPA, so it does not allow AMPA receptors to be endocytosed. And then, of course, potassium channels, in which illustrated here, we show that pKa phosphorylation of potassium channel will lead to their opening. So this was CMP as a second messenger. This is just to remind you that we have other second messengers, such as deacylglycerin IP3. So we know that phospholipase C. So now at the activation of phospholipase C with the GQs, G protein couple receptors then activate GQ, will lead phospholipase C beta activation, which will then hydrolyze phosphonidyl diphosphate into diacylglycerol, which stays in the membrane and can then activate PKC, and also produces inositol 3-phosphate, which will then go and bind its inositol 3-phosphate receptor and in the plasmatic reticulum, which will then lead to the release of calcium from in the plasmatic reticulum. So you will have calcium as second messenger and also have diacylglycerol. So elevated calcium. Calcium is a very interesting second messenger because it is also ion that can go through the channels, but it also is a second messenger, and it's also kept in a very low levels inside of the cells. Remember when we talked about vesicles? So you have a really small, low concentration of 100 nanomoles, 50 to 100 nanomoles inside of the cells, and then you can very quickly, through many signaling pathways converging, can increase the cytosolic calcium concentration from those low amounts to even 0.5 to 1 millimolar. Now, this calcium can exert its effects directly. There are different calcium binding proteins, so it can bind the channels, it can bind enzymes. However, it can also, lots of its effects are exerted through binding to calmodulin. So we're going to spend some time describing what calmodulin here, but this is just a slide to remind you how PLC beta will catalyze cleavage leading to calcium release from the endoplasmatic reticulum. Now, as I said, the predominant mediator of calcium action is calmodulin. Calmodulin is it's really small protein, only 17 kilodalton. It has this dumbbell shape, so it has the alpha helix, and on both sides it has like helical region, spheric region, and at those ends, those spherical regions, which have few alpha helices as well, is where calcium binds. It has a really high calcium affinity, and calcium binding will cause the conformational change and expose different residues that will promote interaction with the target proteins, and those target proteins can include phosphodiesterases, adenylyl cyclases, calcineurin, which is phosphatase, kinases. So it changes its conformation, exposing different residues, then allow it to interact with different proteins. So one of the most common thoughts when you talk about calmodulin and calcium are calcium calmodulin dependent protein kinases, otherwise known as CAM kinases. They're a group of kinases, again family. So we have CAM kinase 1, 2, 3, 4. And they are activated in different way, which is called in this schematics. Pretty much calcium ions are going to bind to calmodulin, and it will activate CAM kinases by displacing the inhibitory domain from the catalytic side. So again, similar to the pKa, and this is going to be the same for pKc and pKg that we're going to be talking about. The activation is really similar. All of these kinases have catalytic domain, and then they have inhibitory domain. That inhibitory domain folds over, and it's inhibiting the catalytic sites, preventing the binding. And activation almost always occurs by displacing that inhibitory domain. In pKa, those are different, so we have different subunits. In the other ones, such as calcium-calmodulin-dependent kinases and pKc, it's the same subunit. So both regulatory and inhibitory domain and catalytic domain are on the same subunit. And then again, here shown is that it leads to direct activation of CAM kinase 3, which can then play a role in the translation, can regulate translation at the level. It can lead to CAM kinase 4, which is the one that often moves into the nucleus, and CAM kinase 2. CAM kinase 2 again has several genes. Some of them are also capable of moving into the nucleus. CAM kinase 2 is the most neuronal abundant, so we're going to spend a little bit more time talking about it. Again, here's the schematic that I just told you. In this case, we have both catalytic domain and regulatory domain in the same subunit, and it has also association domain. So this association domain allows CAM kinase 2 to form this 12-mer. So there's 12 subunits forming. And what's really interesting that when calcium calmodulin binds, it is going to lead to autophosphorylation, but only when it's bound to two neighboring subunits, which is illustrated in B. So you have calcium calmodulin bind to two neighboring units, and then they are going to autophosphorylate each other. And this autophosphorylation allows calmodulin to be independent of calcium calmodulin. And this again depends on the frequency. And how can it depend on the frequency? So again, in this slide, in the top, you have this idea that you need these subunits, both of these neighboring subunits, to bind calcium calmodulin to induce this autophosphorylation that allows a prolonged activity. Now, this autophosphorylation allows prolonged activity because it prevents the binding of inhibitory domain to catalytic site. So if we have a low frequency events, as shown in the beginning, so we have in the pink and the red are both activated, the one that have binding calcium calmodulin, but the red one is the one that's phosphorylated and pink just has bound calcium calmodulin. So in the low frequency, these subunits that bind calcium calmodulin may be able to phosphorylate the next subunit, but then they're going to get off, so you will not be able to propagate this. However, when you have a high frequency event, there will be so much calcium coming in and then so much calmodulin activation that there will be sufficient amount of time for this subunit to phosphorylate each other and then to become independent of calcium calmodulin, which again is allowing maintenance activity even though the signaling is not coming anymore. Now, what I wanted also to talk very briefly here is one of the experimental systems by which you can imagine imaging calcium kinase activation, because this is really a beautiful FRET system in which you can express transgenic protein, CAM kinase 2, or you can make it a knockin, in which you will use FRET system to be able to follow with activation. So normally CAM kinase is folded, these subunits are close to each other when it's inactive, so you will have FRET. So if you use wavelength of one light, you will get the emission of the other wavelength, telling you how much FRET you have. However, when it's activated, this phosphorylation displaces and separates these two subunits that play a role in the FRET signaling far from each other, so you will not have any more FRET. And that is illustrated on the bottom. So the way that CAM kinase was here activated was by inducing local uncaging of the glutamate. So in the panel A, we have local engagement of glutamate leading to increase in calcium concentration, and that's shown using calcium-sensitive dye, fluoro-4, which gives you a green signal, and you can see that that green signal very quickly is induced in these spines, and then by 160 milliseconds pretty much gone. On the other hand, we also have LTP in the stimulated spine. You will have increased activity with the frequency, this engagement of glutamate. However, on adjacent spine, which did not have this glutamate uncaging, you will not have potentiation. And then you can look at CAM kinase activation. It is exactly the same spine in which you can look at the calcium. So again, you can see that using FRET, you will have the red signal, then you will lose the signal, so you will just have you will not be able to transfer, so you will have a decrease in GFP signaling when it is activated. So at 4 and 12 we have activation, and 12 a little bit, and then 30 and 36, 60 and 80 seconds it is going back to normal because we are seeing more of GFP. Okay, so we are now going to talk more briefly about protein kinase C. Protein kinase C is really important because it modulates and it plays a role in exocytosis of synaptic vesicles, which is shown in this panel here. You can see how many of these protein on synaptic vesicles or on the plasma membrane that play a role in the docking and fusion. They're really regulated by both CAM kinases, PKA and PKC. You can see that PKC regulates pretty much most of these targets and the plasma membrane, but also many targets on the surface of the vesicles. And also on the right on the panel, the schematic is supposed to show you that PKC activation is as well going to lead to the changes in the transcriptional factor. In this case, it's not CRAB, it's mostly regulated through activation of AP1, which are composed of CFOS and CJUN. A typical kinase. So the way that PKC was thought to maintain its activity despite the end of the signaling was through PKM zeta formation. So PKM zeta was thought to be formed by proteolysis of active PKC, so we have significant amount of signaling or high frequency stimuli. There will be a protease that's going to cut PKC, so it's going to remove regulatory subunits, allow just catalytic subunits, which will be then PKM zeta. What we know now is that there is no cleavage, but actually what is going on, there is a change in the formation from the internal promoter, so it's actually through from the gene expression. Now we will use alternative internal promoter within PKC zeta gene to make PKM zeta, and that is a form again of learning that will allow PKM zeta activity, PKC activity to be maintained despite the end of the signaling. Now, calcium calmodulin will not only activate calcium calmodulin kinase, or calcium or PKC directly. Calcium calmodulin also will activate NO synthase, nitric oxide synthase. So again, we have signal-to-G protein-linked receptors, IP3 binding to endoplasmic reticulum, increasing the calcium and calmodulin, which then activate nitric oxide synthase, which will produce nitric oxide. So why is this important? It's actually very important in two different systems. One is dilation of blood vessels. We have this nitric oxide, which is now the gas and can then very easily go into the next cells. It can go through the membranes into the next cell. And the one that's produced in the neurons, endothelial cells, in this example, can then cross to the smooth muscle cells on the blood vessel wall, where they will activate guanylyl cyclase. So nitric oxide activates guanylyl cyclase, which will produce cyclic GMP, activation of PKG, and muscle relaxation. And here's PKG shown structure on the panel on your right. Again, it has regulatory and catalytic subunit, and also has inhibitory domain, pseudosubstrate domain, and the hinge that allows it to fold on, so the catalytic subunit is inhibited by its inhibitory domain, and also can dimerize. So PKG actually has dimerization domain. PKG is really crucial for the functioning of Purkinje neurons. So nitric oxide synthase produces NO in the parallel fibers, and this nitric oxide will then go through the plasma membrane into Purkinje neurons. There it's going to activate guanylyl cyclase, leading to increased level of cGMP, and activate PKG. Now PKG, when it's activated, it's going to affect the activity of phosphatase, and that phosphatase is actually known to prevent endocytosis of AMPA receptors. So PKG, by inhibiting this phosphatase activity that prevents endocytosis, will actually stimulate AMPA receptor endocytosis and lead to reduced expression of AMPA receptors on Purkinje neurons and form of LTD. So in this problem is what I want you to think about at home. So you have a hypothesis, which is shown below, is that this adapter protein called SEP97 will bind to the C-terminus of AMPA receptor via PDZ domain. And it's going to bind to ACAP79 through SH3 domain and GK domain. So these are different protein domains. You don't need to know exactly what they do, but those are the domains through which SEP97 binds either to AMPA receptors or through ACAP79. This binding will form a complex or scaffold that positions PKA, which binds ACAP79, near AMPA receptor, where it can phosphorylate at the A45 and allow it to stay in the membrane. Now, how would you test whether interactions of SEP97 and ACAP79 and, on the other hand, SEP97 and AMPA receptor are required for phosphorylation of AMPA here in A45? So I want you to think about the experiment that you would do